Well, hello and welcome to the JASN podcast, the monthly podcast where we highlight an exciting paper from the current issue of the journal. These papers may be clinical, basic, or translational. I am Dr. David Ellison, professor of medicine and chemical physiology and biochemistry at Oregon Health and Science University and a deputy editor of the journal. And I'm joined today by Professor Andrew Hall from the University of Zurich to discuss his paper entitled Spatiotemporal Landscape of Kidney Tubule Responses to Glomerular Proteinuria, which appeared recently in JASN. Professor Hall, if you could introduce yourself and tell us a little bit about your background.
Sure. Well, firstly, thank you very much for the invitation. So I'm originally from the UK. I trained in nephrology there. And as you say, I'm now a professor at the University of Zurich. I'm also the director of the Zurich Kidney Center, which is a research and education network within Switzerland. And my main scientific interest is the proximal tubule, the physiology and diseases of the proximal tubule. And the paper, the study we're going to discuss today was a collaborative effort between my group in Zurich and also the groups of Sophie de Senya in Geneva and Pietro Chippa in Lugano.
Great. Well, let's dive right in. So your paper uses single nuclei RNA sequencing and other techniques to analyze the effects of proteinuria on tubule phenotype. For these studies, you used the POTATAC model to induce proteinuria. So before we get into the meat of the paper, perhaps you could tell us a little bit about this model and what its advantages are and what its limitations are. And one specific question I wanted to ask you is, do mice, when you induce the proteinuria, do they develop hypoalbuminemia like a nephrotic patient would?
Sure. Well, this model was developed about 10 years ago by another group, and I think it does quite closely reflect human nephrotic syndrome. So to answer your specific question, yes, the mice do get a decrease in blood albumin. They get albuminuria, they get edema. So really, they do get the features of human nephrotic syndrome. I guess one thing that's perhaps a little bit different from humans is the speed of onset. It's a very fast onset. Within a couple of days, these mice have, really striking proteinuria. Nephrotic syndrome in humans, as you know, is typically more insidious. But from an experimental point of view, it does allow us to see how tubules respond to quite a rapid increase in protein filtration.
Great, thank you. So much of your work focuses on the proximal tubule, which you just said is an area of special interest for you. And even before you induce proteinuric injury, you make several interesting observations regarding properties of the proximal tubule. Although I was aware that SGLT2 predominates in the S1 segment of the proximal tubule, I personally had not understand how much organic anion transport pathways and secretory pathways generally were more restricted to later proximal segments. So can you tell us what you see as the most important new information about normal proximal tubules to come out of your work?
Yes, I think the sort of take-home message is that we'd underestimated just how much heterogeneity there is along the proximal tubule. I mean, in your field of the distal tubule, we clearly segregate the different segments. We understand they're functionally very different. In the proximal tubule, we often tend to lump all the segments together in studies. And I think the more you look, whether it's with single cell sequencing, whether it's with functional imaging, the more you look, the more you really see differences along the proximal tubule. I think we're still understanding really what those differences are and what they mean and what the factors are that shape them. But clearly there are big differences occurring as we go along the proximal tubule. And I think this is something that we had underappreciated, but is now becoming more clear.
Yeah, well, one obvious follow-on question to that is that when you induce proteinuria, you find that cells in the S2 segment change to become more like those in S1, and that cells really look like they become more able to reabsorb protein like the S1 cells. And this makes some sense, as more distal cells are likely participating in more endocytosis than they are normally when you induce the proteinuria. So one thing that's characteristic of the S1 segment normally is a very highly developed brush border, which tends to reduce as you go along the length of the proximal tubule. Do you see evidence that this becomes more well developed after you induce the proteinuria?
Well, it's a great question, but unfortunately, it's not something we specifically looked at. I think to really answer that in detail, one would have to do electron microscopy, and this is not something we did. What we can say is for sure, there's a lot of development of the endolysosomal system, a lot of increased expression of endolysosomal proteins in the S2 segment in under proteinuric conditions. So it would be very interesting to look at the brush border to see if this also develops in parallel. But at the moment, unfortunately, we don't have the answer to that.
So why do you think what do you think signals the S2 segments to lose some of their secretory features when you induce the proteinuria state? Do you think they just get sort of distracted by all the protein they need to absorb? Your observation about reduced lipid metabolism and transport more distally in nephrotic mice might contribute to the the lipid abnormalities we see in nephrotic patients. I think that's quite an interesting suggestion. And could you explain that as well? What is the role of the proximal tubule in lipid metabolism normally? I guess these are several questions that you might speculate about.
Okay, well, to take the first question, why are we seeing a downregulation of normal S2 functions? I think there's two things going on. Firstly, we see a remodeling towards, or partial remodeling towards an S1 phenotype. Secondly, we see the development of injury and dedifferentiation. And when we do a lineage analysis, we can see that's the remodeling that seems to be happening first, and then the damage and dedifferentiation happening second. What the relative contributions of those are to losing canonical functions in S2, we couldn't really say at this point. I suspect it's probably a combination of both. Coming to the second point about lipid metabolism, so it's become a sort of dogma in the field that the proximal tubule predominantly relies on lipids as its fuel, and I'm sure that's true to an extent. But if you look along the proximal tubule, there are big differences in metabolism between S1 and S2. We see a much higher expression of lipid-related genes in S2. We see lipid-containing structures in S2, like multilamellar bodies. We see a much higher density of things like peroxisomes in S2, which are obviously very involved in lipid metabolism. So there's a lot of strands of evidence that there may be more lipid metabolism happening in S2 than S1 for reasons that we don't yet fully understand. And then coming to the nephrotic syndrome, obviously, as you say, we see huge changes in systemic lipid homeostasis in patients with a nephrotic syndrome for reasons I don't think are fully explained as yet. But one could speculate that if we're normally transporting lipids in S2, and certainly from the gene expression data, it seems that lipid transporters are quite highly expressed in S2. Then if we lose that expression, then one could speculate that could have a knock-on effect on systemic lipid metabolism. So that's one possibility, but we obviously don't as yet have direct evidence for that.
And you probably said this in the paper, but are the lipids filtered and then absorbed from the apical side, or are they mostly taken up basolaterally?
Well, that's a really good question. I don't think we know. There's still some controversy about where exactly the lipid transporters are, if they're more apical or basolateral. It could also depend on the lipid and in what form it is, whether it's in a triglyceride form, whether it's bound to proteins, whether those proteins are filtered or not. I think there's a lot of variables there. And I don't think we understand that well enough, but I think it's a really important question.
So another observation that I thought was pretty cool that you make is by comparing the results of these studies with proteinuric mice with ischemia reperfusion. And as I understand it, in the latter, in the ischemia reperfusion mice, injury and dedifferentiation is pretty much universal across the proximal tubule. But in the proteinuric mice, the injury and dedifferentiation is the most prominent beyond the S1 segment. It's as if the S1 is already reabsorbing proteins and therefore more protein exposure doesn't bother it. Is that a reasonable way of looking at this? And maybe you could comment some more on the differences between the different models.
Yeah, I think I would agree with that. So we know the proximal tubule is exquisitely sensitive to ischemia, and in previous single cell studies, it's been shown that there is a rapid dedifferentiation of all three segments of the proximal tubule, which then recovers fairly quickly, as we know. Whereas in this study, where we looked at the effects of proteinuria, it was really the S2 and S3 that were showing more injury markers, not only at RNA level, but also when we did antibody staining for injury markers, we really saw them mainly in S2 and S3. We don't fully understand the reasons for that. We did, a year or two ago, we published a paper showing that after proteins are metabolized in S1, they are to some extent secreted and then reabsorbed further down the tubule. So one could imagine that in a proteinuric state, that if the S1 is reabsorbing, degrading, and secreting more proteins, this could cause a peptide overload down in S3. So this could be one possibility. As you say, our data suggests that S1 was relatively well protected from this proteinuric state, presumably because it has already such a high capacity to process proteins. And interestingly, although we lost S2 markers in the proteinuric mice, S1 markers like SGLT2 were relatively well preserved. And from a translational standpoint, if this was also the case in humans, then, it might raise the question as to whether SGLT2 inhibitors could be useful, perhaps for treating the fluid overload that occurs in patients with nephrotic syndrome, particularly given that they tend to become resistant to conventional diuretics that are targeting the distal part of the nephron.
Yeah, SGLT2 inhibitors seem to be useful in every kidney and cardiovascular syndrome, so it wouldn't be surprising. So we normally think of albumin as the primary protein that we filter in nephrotic syndrome, but obviously there are other proteins as well. As you know, one model for diuretic resistance is that filtered proteases can activate ENaC distally and thereby lead to primary sodium retention. Do you think that albumin is the primary bad actor in this in the proteinuric mice, just based on its sort of the predominance of its magnitude of filtration, or do you think some of the effects may be related to other proteins that are filtered as well?
Yeah, so I mean, as you say, albumin obviously will be the predominant protein here, but it will then compete for uptake in the proximal tubule with other proteins that are normally reabsorbed in the proximal tubule and that would not appear normally in the distal part of the nephron. And some of these proteins, these small proteins, are quite bioactive, small hormones, etc. So one could speculate that if these start appearing in the later parts of the nephron, they could have signaling effects, and this could be a possibility. But obviously, from this study, we don't have any direct evidence for that.
So maybe I'll just ask one last question. As you note, my own laboratory is focused primarily on the distal tubule, and we've also used RNA-seq to study distal tubule injury models, although not proteinuria. You report that distal cells also dedifferentiate in your model, losing some NKCC2 along thick ascending limb and NCC along the distal convoluted tubule, two proteins that have long been targets of our own work. We also see characteristic features, including a loss of proteins that normally characterize distal tubule cells when they're remodeling. So they often remodel by losing some of these typical epithelial proteins when they remodel. Given that the cells in the distal tubule don't have a lot of endocytic machinery and certainly not there primarily to do endocytosis, what do you think it is about the proteins entering the distal tubule that stimulates them, or what are they actually becoming endocytic, and is that part of the injury process?
Yeah, so I mean, what we saw in this study was that, as you say, that all of the segments of the distal tubule seem to be dedifferentiating and showing genetic convergence in response to proteinuria. In terms of what stimulates this, so as you say, they don't normally endocytose, but one could imagine if the concentration of protein becomes very high in the lumen of the distal tubule, which it will be with the urine being concentrated by that point, one could imagine that some endocytosis might occur. But I'd also go back to my previous answer where I said we have some bioactive proteins here that maybe could activate signaling mechanisms in the distal tubule. This could be a possibility too. But what we saw with antibody staining was that the major targets of diuretics in the thick ascending limb, the distal convoluted tubule, were being downregulated in the proteinuric mice. And in combination with this loss of S2 secretion, I mean, this could certainly explain diuretic resistance, because these diuretics have to be secreted by the proximal tubule to enter the lumen of the distal tubule and then hit these apical transporters. So perhaps it's a combination of these processes that are leading to this diuretic resistance. But in terms of what actually causes the dedifferentiation of distal tubule, I think it's still an open question.
Yeah, I thought that was a fascinating speculation about potential contributors to diuretic resistance. As you know, there's been a huge literature and long speculation about why nephrotic patients seem to be resistant, and it probably is multifactorial, but downregulation because of dedifferentiation in the distal nephron. I thought that was quite provocative, and I like that suggestion. So I think we're out of time, and I really, really enjoyed reading your paper. I think it raises lots of questions, which is what good papers often do. And I hope you're working to follow up on some of the other aspects of how proteinuria can damage the kidney. Thank you so much.
Thank you. Thank you for highlighting our study. I appreciate it.
This podcast is copyrighted by the American Society of Nephrology. All rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare professional if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.